In this presentation, I am delivering a lecture on an introduction to biomolecules. The agenda of my presentation is given here: biochemistry, an interdisciplinary subject; biomolecules, an introduction; carbohydrates; amino acids and proteins; lipids; nucleic acids; and biomolecules, conclusion. Biochemistry is an interdisciplinary subject that integrates multiple perspectives from different disciplines such as chemistry and biology. Biomolecules is a basic subject under biochemistry in which the different aspects of biology, chemistry, and physics of the chemical molecules present in the living organisms, as well as their interrelationship, will be taught to the students. Biochemistry is the field of study concerned with chemical substances and the processes that occur in the living organisms. Chemistry is the study of chemical structures and properties of all the materials on the planet and involves more mathematical concepts. Biology is the study of living things and biological systems, animals, plants, and microorganisms, and involves very less mathematical concepts. Biochemistry is the study of structure and properties of the chemical compounds present in the living organisms on the planet and involves some mathematical concepts. In simple terms, biochemistry is a combination of chemistry and biology. It is the chemistry of life. There is structural hierarchy of chemical molecules in biochemistry: atom, molecule, macromolecule, organelle, cell, tissue, organ system, and organism. The examples for each of these are given here. While chemistry deals with more than 100 elements, the 6 major elements in biochemistry are carbon, hydrogen, nitrogen, oxygen, phosphorus, and sulfur. These elements constitute 98% of the biomolecules. There are also other elements such as calcium, iron, copper, zinc, iodine, magnesium, sodium, potassium, chlorine, manganese, selenium, cobalt, etc., which are present in minute quantities and are called as trace elements. The important molecules are monosaccharides, disaccharides, amino acids, fatty acids, glycerol, nucleotides, etc. The important macromolecules are starch, glycogen, proteins, lipids, and nucleic acids. Nucleus, mitochondria, chloroplast, endoplasmic reticulum, microsomes, and lysosomes are some of the important cellular organelles. There are 4 types of cells: epithelial cells, nerve cells, muscle cells, and connective tissue cells. Epithelial tissue, nerve tissue, muscle tissue, and connective tissue are the 4 types of tissue. The important organs are brain, liver, heart, lungs, pancreas, gallbladder, and kidney. There are different organ systems such as nervous, digestive, respiratory, circulatory, and excretory systems. The examples for organisms are humans, plants, animals, bacteria, fungi, algae, and virus. While chemistry deals with atoms and molecules, and biology deals from organelles to organs, biochemistry deals from molecules to organ systems, with macromolecules as the unique subject for biochemistry. Biomolecules are the important molecules present in the living system. In this section, we will learn about these molecules. Biochemistry can be classified based on the organic or inorganic nature of the chemical molecules into 2 branches: bioorganic chemistry and bioinorganic chemistry. Biomolecules comes under bioorganic chemistry. There are 2 groups in biomolecules: major and minor. The carbohydrates, proteins, lipids, and nucleic acids are the major biomolecules, while vitamins and hormones are the minor biomolecules. Biomolecules can also be classified into micromolecules and macromolecules based on their size, molecular weight, and solubility. The micromolecules are small in size with their molecular weight less than 1000 and are not precipitated by trichloroacetic acids. Minerals such as sodium, potassium, chlorate, and calcium, sugars like glucose, fructose, sucrose, lactose, and maltose, amino acids such as glycine, tyrosine, aspartic acid, and glutamate, fatty acids like palmitic acid, oleic acid, linoleic acid, and acetic acid, and nucleotides such as ATP, GTP, CMP are the examples for the macromolecules. The macromolecules are large in size with their molecular weight more than 1000 and are precipitated by trichloroacetic acid. Carbohydrates like starch, glycogen, and cellulose, proteins like collagen, keratin, and growth hormone, lipids like triglycerides, HDL cholesterol and oils, nucleic acids like DNA, RNA, that is mRNA, rRNA, and tRNA, and hormones like HCG, glucagon, and TSH are the examples for the macromolecules. Biomolecules are chemical compounds containing carbon, hydrogen, nitrogen, and oxygen as their major elements. They may be monomers, oligomers, or polymers of the smaller building blocks or micromolecules. These biomolecules are the vital components of the organisms. The polysaccharides are made up of multiple units of monosaccharides, and their major functions include energy storage, support to cell membranes, and as recognition molecules in immune function. Proteins are composed of amino acids and are responsible for maintenance of structure of organelles to organisms, catalysis of reaction, cell signaling, recognition of signaling, and transport of solutes across the cellular membranes. DNA is the polymer of deoxyribonucleotides and is responsible for storage and transmission of hereditary information. RNA is the polymer of ribonucleotides and is involved in protein synthesis. Now we will discuss about the carbohydrates. The classical definition of carbohydrate is that they are the hydrates of carbon. If three elements, carbon, hydrogen, and oxygen, are present in a ratio of about 1:2:1. The main functional groups of carbohydrates are either aldehyde or ketone along with hydroxyl group. The general formula for monosaccharides is CnH2nOn, and for other carbohydrates is CxH2OY, with the value of Y usually less than the value of X. Based on configuration at the penultimate carbon, the carbohydrate exists in two forms, D form and L form, and only the D form is present in the living organisms. The modern definition for carbohydrate is that they are optically active polyhydroxyaldehydes or polyhydroxyketones, or compounds which produce these on hydrolysis. The structure of the carbohydrates can be drawn as linear open chain form and as closed ring structure in 6-membered pyranose form or 5-membered furanose form, depending on the number of atoms involved in the ring formation. In the closed ring form, they can exist in two forms, alpha form and beta form, based on the position of the OH group in the first carbon. These compounds are optically active. Based on their optical rotation, the monosaccharides exist in two forms, D form dextro and L form levo. Both the dextro forms and levo forms are present in the biological system. For example, D-glucose is dextrorotatory and is known as dextro. Similarly, D-fructose is known as levulose since it is levorotatory. Carbohydrates can be classified on the basis of the number of sugar units that are present in a particular carbohydrate. Thus, carbohydrates can be classified into three types: monosaccharides, oligosaccharides, and polysaccharides. Thus, the carbohydrates having a single sugar unit which cannot be hydrolyzed any further are known as monosaccharides. These are the basic units for this classification. As we have seen already, the monosaccharides may have aldehyde or ketone as their functional group. These monosaccharides with aldehyde functional groups are called as aldoses, and those with ketone functional groups are called as ketoses. Similarly, based on the number of carbon atoms in the monosaccharides, they are called as trioses, those with 3 carbon atoms, tetroses, 4 carbon atoms, pentoses, 5 carbon atoms, hexoses, 6 carbon atoms, and so on. All the monosaccharides are also known as reducing sugars, as they exhibit reducing property by reducing copper in the Benedict's reagent or filling solution. The examples for these sugars will be given little later. The carbohydrates made up of 2 to 10 sugar units are called as oligosaccharides. These can be hydrolyzed into constituent monosaccharides. Depending on the number of monosaccharide units, the oligosaccharides can be classified into disaccharides made up of 2 monosaccharide units, trisaccharides, 3 monosaccharide units, and so on. The carbohydrates made up of more than 10 sugar units are called as polysaccharides. They can be hydrolyzed further into oligosaccharides and monosaccharides. Based on the composition of monosaccharides in them, the polysaccharides can be classified into homopolysaccharides, polymer of same monosaccharide unit, and heteropolysaccharides, polymer of different monosaccharide units. The structure of the all those monosaccharides are given here. Trioses, tetoses, pentoses, and hexoses are represented here. The names of the metabolically important monosaccharides are marked in boxes. Glyceraldehyde is an intermediate in the glycolysis process. Erythrose is an intermediate in HMP pathway and photosynthesis. Ribose is a component of nucleic acids. Arabinose is present in glycoproteins, while xylose and mannose are present in proteoglycans. Glucose is the most important monosaccharide unit as it is universally present in almost all foods and all metabolic pathways. Galactose is present in lactose, the sugar present in milk. Here the structure of the ketose monosaccharides are given. Dihydroxyacetone is an intermediate in the glycolysis process. Erythrose is involved in tanning process in the skin. Ribulose and xylose are intermediates in HMP pathway and photosynthesis. Fructose is present in honey, many fruits, and is present in sucrose, the common sugar. Disaccharides are made up of two monosaccharide units. Maltose contains two glucose units and is present in germinating grains and many plants and vegetables. Sucrose, common sugar, contains glucose and fructose and is present in sugar cane. Lactose is made up of glucose and galactose and is present in milk. Raffinose is a trisaccharide made up of one unit each of galactose, glucose, and fructose and is present in beans, cabbage, vegetables, and whole grains. Of the polysaccharides, starch and cellulose are the most important plant polysaccharides. Starch is the storage form of carbohydrates in all the plants and is the main source of energy for us in the form of food. Cellulose is present in the cell walls, providing protection to the plant cells and is used as food by ruminants and other animals, but not by humans, as humans cannot digest and metabolize cellulose. Glycogen is the major carbohydrate present in the animals and is the storage form of carbohydrates. Starch, cellulose, and glycogen are the polymers of glucose and are examples for homopolysaccharides. Of the heteropolysaccharides, the structure of chondroitin is given, which is a mucopolysaccharide containing nitrogen and sulfur moieties also. Agarose and carrageenan are the heteropolysaccharides present in the algae. The carbohydrates are the main source of energy for almost all the organisms. In the human body, brain cells and RBCs are almost wholly dependent on carbohydrates as the energy source. On digestion and metabolism, the consumed carbohydrates are converted into glycogen and the excess amount is converted into fat for storage. Carbohydrates are also precursors for other biomolecules such as amino acids and lipids through integration of metabolic processes. They are also present in glycoproteins and glycolipids in the cell membrane and receptors. They are also involved in various functions like cell growth and fertilization. They are also present as structural components like cellulose in plants, exoskeleton of some insects, cell wall of microorganisms, and mucopolysaccharides as ground substance in higher organisms. They are stored in animals as glycogen and in plants as starch to meet their energy demands. Our next biomolecule will be amino acids and protein. Amino acids contain carbon, hydrogen, nitrogen, and oxygen, and some amino acids contain sulfur also. They are carboxylic acids having an amino group at the alpha position. Thus, they have two functional groups, carboxylic group and amino group. They have the unique property of existing in Jupiter ionic form, that is a molecule having both positive charge due to amino group and negative charge due to carboxylic group. Based on configuration at the penultimate carbon, amino acids exist in two forms, D form and N form, and only the L form is present in the proteins of the living organism. The general formula for amino acids is RCHNH2COOH, or can be hydrogen, hydrocarbon chain, or a cyclic group. Based on the nature of this R group, amino acids can be classified into 5 groups. Non-polar aliphatic amino acids, in which the R groups are non-polar and hydrophobic, such as methyl group. Aromatic amino acids, in which the R groups are aromatic side chains, such as phenyl group, and are relatively non-polar, hydrophobic. Polar uncharged amino acids, in which the R groups are more soluble in water and are hydrophilic because they contain functional groups that form hydrogen bonds with water, such as hydroxymethyl group. Positively charged basic amino acids in which R groups have significant positive charge due to the presence of a secondary primary amino group in lysine or a positively charged guanidino group in arginine or an imidazole group in histidine, and negatively charged acidic amino acids in which R groups have a carboxyl group. In this slide, the structures of the nonpolar aliphatic amino acids having aliphatic linear or cyclic side chain and aromatic amino acids having aromatic rings as the side chains, and polar uncharged amino acids having linear aliphatic polar side chain, positively charged basic amino acids having positively charged amino group or guanidino group or an imidazole group as the side chain, and negatively charged acidic amino acids having additional carboxyl group as the side chain are represented. Proteins are the polymers of L-alpha amino acids. They are the most abundant organic molecules in the living system and form about 50% of dry weight of cell in almost all organisms. Whether they are of animal origin, plant origin, or microbial origin, these proteins contain the 20 standard amino acids in the structure. The proteins are very important for the architecture and functioning of the cell. Collagen, a protein present in the muscle, is the most abundant protein in animals, while rubisco, an enzyme involved in photosynthesis, is the most abundant protein in the plants. The synthesis of protein is controlled by DNA and facilitated by RNA and enzymes. Proteins can be classified on the basis of composition and function. Based on composition, proteins can be classified into simple proteins, conjugated proteins, and derived proteins. Simple proteins contain only amino acids. They can be further classified into globular proteins and fibrous proteins based on their shape. Globular proteins are somewhat spherical in shape and include albumins, globulins, glutamins, prolamins, histones, globins, protamins, etc. Fibrous proteins are long, rod-like proteins and include collagen, elastin, keratin, etc. Conjugated proteins have additional groups apart from amino acids. Based on these additional groups, they can be classified into nucleoproteins having nucleic acids, glycoproteins and mucoproteins having carbohydrates, lipoproteins having lipids, phosphoproteins having phosphate groups, chromoproteins having chromophores or colored chemicals, and metalloproteins having metals. Derived proteins can be further classified into primary derived proteins and secondary derived proteins. Coagulated proteins, proteins, and metaproteins are the primary derived proteins, while secondary derived proteins comprise proteoses, peptones, polypeptides, and peptides. Based on their biological functions, protein can be classified into contractile or motile proteins that help in the muscle contraction and relaxation, defense proteins that are involved in the protective functions in the bloodstream, catalytic proteins are enzymes that catalyze the biological reactions, nutrient and storage proteins that are stored as reserve food, regulatory proteins are hormones that control the metabolic processes, structural proteins that are involved in giving shape and structure to the organs and organisms, toxic proteins which are used as toxins by some organisms, and transport or carrier proteins which are involved in the movement of various nutrients. Protein structure has 4 levels of organization. The simple amino acid sequence is considered as the primary structure. This involves only the peptide bond. In the secondary structure, the polypeptide chain may be coiled into an alpha helix or arranged in parallel structures known as beta-pleated sheets. Only hydrogen bonding is involved in this level. This structure will be further folded to give the tertiary structure. This process involves many interactions such as electrostatic bond, hydrophobic interactions, disulfide bond, and van der Waals forces. If there are more than one polypeptide chain in a protein, they are assembled into a single unit, and this is called its quaternary structure. This assembly requires interactions like hydrogen bonds, electrostatic bonds, hydrophobic bonds, and van der Waals forces. The functions of proteins include catalytic functions as enzymes, contractile or motile functions, defense functions, genetic functions, nutrient and storage functions, protective functions, regulatory functions of hormones, structural functions, and transport functions. Next, we will discuss about the lipids. Lipids are a heterogeneous group of naturally occurring organic compounds. They include fats, oils, steroids, waxes, and other compounds, which are related more by their physical properties rather than their chemical properties. They are relatively insoluble in water and soluble in non-polar solvents such as ether and chloroform. They are the important dietary constituents in almost all foods. They contain carbon, hydrogen, and oxygen, and also phosphorus, nitrogen, and sulfur. The ratio of C/H is 1:2 in lipids, with the amount of oxygen and other elements being very less. Lipids are the main concentrated storage form of energy, and they form about 3.5% of the cell content. Based on the composition and chemical nature, lipids can be classified into simple lipids, compound lipids, and derived lipids. Simple lipids are the esters of fatty acids with glycerol and other higher alcohol. Compound lipids are fatty acids esterified with alcohol, but in addition they contain other groups. Depending on these extra groups, they are subclassified as phospholipids containing phosphoric acid, glycolipids containing sugar derivative, and sulfolipids containing sulfate groups. Derived lipids are compounds which are derived from lipids or precursors of lipids. Example, fatty acids, steroids, etc. Based on polarity, lipids can be classified as neutral or storage lipids comprising triglycerides, which are the major storage lipids in organisms, and polar or membrane lipids comprising phospholipids and glycolipids. The phosphate groups and sulfate groups confer polarity, and these are mainly present in the biological membranes. Fats can be differentiated into simple fats and mixed fats. Simple fats contain identical fatty acids attached to glycerol, while mixed fats contain 2 or 3 different fatty acids attached to glycerol. Waxes are lipids which are made up of long-chain saturated fatty acids and a long-chain saturated alcohol of higher molecular weight other than glycerol. Examples include beeswax, lanolin or wool fat, sebum, cerumen or airwax, plant wax, and paraffin wax. In phospholipids, glycerol is attached to 2 fatty acids and a phosphate group attached to a nitrogenous base or an alcohol. These groups can be choline in phosphatidylcholine, ethanolamine in phosphatidylethanolamine, serine in phosphatidylserine, inositol in phosphatidylinositol, glycerol in phosphatidylglycerol. Diphosphatidylglycerol has 2 phosphoglycerol groups attached to a glycerol. Cerebroside is a glycolipid present in brain. It has a long-chain fatty acid and a sugar attached to a sphingosine molecule. Sulfolipid contains a sulfur sugar moiety attached to a diacylglyceryl group. Fatty acids are carboxylic acids. They can be of 2 types, saturated fatty acids and unsaturated fatty acids. Saturated fatty acids have only single bonds in their aliphatic chain. The biologically important saturated fatty acids are formic acid and stearic acid. The unsaturated fatty acids have double bonds in their aliphatic chain. Based on the number of double bonds, they are called as monounsaturated fatty acid or monoenoic fatty acid, dienoic fatty acids, and more importantly, polyunsaturated fatty acids. Other derived lipids include alcohols and sterols. Glycerol and sphingosine are the most common alcohols in the lipids. Cholesterol is the most common animal sterol and is a precursor of various hormones such as aldosterone, cortisol, corticosterone, progesterone, estradiol, testosterone, etc. Plant sterols are known as phytosterols. Triglycerides are the major storage form of energy as the fuel reserve of the body. Phospholipids and cholesterol function as the structural components of biological membranes and regulate the membrane permeability. Cholesterol is the source for vitamin D and also help in the absorption of fat-soluble vitamins. Steroid hormones, prostaglandins, and thromboxanes are important cellular metabolic regulators. Amphipathic lipids such as phospholipids and salts of triglyceride act as surfactants, detergents, and emulsifying agents. Triglycerides act as electric insulators in neurons. Lipids play an important role in giving shape and contour to the body. They also protect internal organs against changes in external temperature and also improve taste and palatability of food. Our next biomolecules will be nucleic acids. The nucleic acids are the molecular repositories of genetic information. They store and transmit genetic information from one generation to the next generation, thus performing a fundamental condition for life propagation. They contain carbon, hydrogen, nitrogen, oxygen, and phosphorus. There are two types of nucleic acids: DNA or deoxyribonucleic acid and RNA or ribonucleic acid. The RNA are further classified into three types based on their function as messenger RNA, ribosomal RNA, and transfer RNA. These nucleic acids are polymers of nucleotides or simply polynucleotides. There are two types of nucleotides which are known as ribonucleotides and deoxyribonucleotides. A nucleotide is made up of three components. They are a nitrogenous base, a purine or a pyrimidine, a pentose sugar, either ribose or deoxyribose, and phosphate groups esterified to the sugar. The purine molecules are adenine and guanine, and the pyrimidine molecules are cytosine, thymine, and uracil. While adenine, guanine, and cytosine are present in both DNA and RNA, thymine is present only in DNA and uracil is present only in RNA. Of the pentose sugars, deoxyribose is specific for DNA and ribose is specific for RNA. The structural details of the purines, pyrimidines, and sugars are given here. Also, a representative picture of ATP, adenosine triphosphate, an important nucleotide for nucleic acid biosynthesis, as well as the energy currency in metabolic reaction is also given. The diagrams of the deoxyribonucleotides and ribonucleotides are given here. The Watson-Crick model of DNA structure is described here. The base sequence is always written from the 5' end to the 3' end. DNA consists of two polydeoxyribonucleotide chains twisted around one another in a right-handed double helix similar to a spiral staircase. The sugar and phosphate groups comprise the handrail, and the bases jutting inside represent the steps of the staircase. The two strands are always complementary to each other, so the adenine of one strand will pair with thymine of the opposite strand, while guanine will pair with cytosine. This base pairing is based on Chargaff's rule. The DNA strands are held together mainly by hydrogen bonds between the purine and pyrimidine bases. There are two hydrogen bonds between adenine and thymine, while there are three hydrogen bonds between cytosine and guanine. The GC bond is therefore stronger than the AT bond. The two strands in a DNA molecule run anti-parallel, which means that one strand runs in the 5' to 3' direction, while the other is in the 3' to 5' direction. In the DNA, each strand acts as a template for the synthesis of the opposite strand during replication process. The spiral has a pitch of 3.4 nanometers per turn. Within a single turn, 10 base pairs are seen. Thus, adjacent bases are separated by 0.34 nanometer. The diameter or width of the helix is 1.9 to 2 nanometers. Messenger RNA or mRNA acts as a messenger for the information in the gene in DNA to the protein synthesizing machinery in the cytoplasm. It carries the message to be translated to a protein. The mRNA is a complementary copy of the template strand of the DNA. Ribosomal RNA is a component of the ribosome which provides the necessary infrastructure for the mRNA, tRNA, and amino acids to interact with each other for the translation process. Thus, ribosomal assembly is the protein synthesizing machinery. The transfer RNA show extensive internal base pairing and appear clover leaf-like structure. They contain a significant proportion of unusual bases. These include dihydrouracil, pseudouridine, and hypoxanthine. Moreover, many bases are methylated. They transfer amino acids from cytoplasm to the ribosomal protein synthesizing machinery. Each molecule is only 73 to 93 nucleotides in length, much shorter than the mRNA molecules. The nucleotides, particularly ATP, are the energy currency in metabolic transactions. Nucleotides are also components of important coenzymes like NAD+, NADP+, FMN, and FAD, and metabolic regulators such as cyclic AMP and cyclic EMP. They serve as donors of phosphoryl groups, ATP or GTP, of sugars, UDP or GTP sugars, or of lipids, PDP acyl glycerol. DNA is the chemical basis of heredity and is a reserve bank of genetic information. It is also responsible for maintaining the identity of different species or organisms over millions of years. Every aspect of cellular function is under the control of DNA. DNA is organized into genes, which are the fundamental units of genetic information. Genes control the protein synthesis through the mediation of RNA. The interrelationship of DNA, RNA, and proteins constitute the central dogma of molecular biology, the central dogma of life. Now we can conclude with a sum up on biomolecules. Biomolecules are important for the functioning of all the living organisms. They also play indispensable roles in all life processes. Carbohydrates are the essential energy sources and structural components of all life. They are the most abundant biomolecules on Earth. Proteins are the major structural elements of cells. They also serve as transporters, enzymes, hormones, receptors, antibodies, etc., which are very essential for the life processes to take place. Lipids are the sources of stored energy. They also act as chemical messengers in the form of steroid hormones. They are the important components in cellular metabolism. Nucleic acids have the unique function of storing an organism's genetic information and transmitting that information from one generation to the next generation. Thus, all the biomolecules are vital for life processes to go on smoothly. Thank you for your attention and patient listening. My contact details are given here. I will be available for any queries on this topic or any related topic.